go around the room. I'm gonna ask, how start over here with Danielle. Ask Danielle a question. Danielle, if you get it right, you get to roll your dice to determine how many points you get. So you need to keep track of your own points. So you have addition first thing in the morning. So add up your points, keep track of them. Again, Jaden and Hayden. If it becomes too much, I will take it away and then you can't play it anymore. Make sure you are focusing. If Danielle doesn't know, I'll just move to the next person in line. So it would be Lana going front to back. So make sure you're paying attention. You're ready to go. Whenever I ask the question, because I'm not going to repeat the question over and over and over again. I'm going to ask it pretty much 1 or 2 times, and that's it. All right. So Danielle, starting over here, what is the science of classification?
Taxonomy.
Good job. Roll your dice. Determine how many points you get.
6.
Good job. So write down 6 on your paper so you can remember how many total points you get. All right, Lana. Who came up with the word species?
John Ray.
John Ray. Roll your dice.
Do I try again?
Yeah, roll again. If it falls on the floor, you get 0 points. Try to get on your dice. Good job. All right. What does species mean, Hayden?
The way we classify things.
Nope.
Species. It's a type of animal, like
Close enough.
cat
Distinct
or-
type of organism. Roll your dice.
3.
All right. Let's see. Who's remembered as the father of taxonomy? What? Nope. Linnaeus. I'll take it. Carlos Linnaeus. Where's Scott?
We don't know.
Okay.
His brother's here.
Okay. Maybe Scott just had a doctor's appointment. All right. Let's see. There are the 2 kingdoms that Linnaeus originally came up with. Name one.
Animalia.
Good job. That's the animals. Okay. Try to roll it off your fingers since I know on the paper it doesn't roll well. There you go. All right, Hayden. What is the other one that he came up with? Oh, I'm sorry. Lana, what was the other kingdom that he came up with? Planty. Roll. What did you on? Keep up with the points. Add them up. All right, there's 4 other kingdoms we have added on today. What's another one?
Phylum.
Oh, okay. So there's 2 kingdoms. One for the plants, one for the animals. What's another one that we've added on to? Because we have more than just plants and animals now. We have a better organizing system. What's the one that has the? Sorry, what?
The fungi.
Yeah, like. So is that your guess?
Yeah.
Yeah, human fungi.
5.
All right, Jaden. What's another kingdom?
Archaebacteria.
You know what's in that one?
Bacteria that fights disease.
Okay, it has.
I have to know what it does.
Yes, but I'll let you roll since you got archaebacteria. These are the bacteria that can survive in extreme environments, like really hot or really salty. Roll.
No.
Oh, 0. Sorry, Jaden. All right, going back over to Glenn. You may stand up to get it really quick. All right, what is another kingdom? So we have Animalia, Plantae, fungi, archaebacteria. What's another one? What's the one that has the regular bacteria in it? Okay, we gotta get. All right, we'll see if someone is any closer than that. Eubacteria. So just put U in front of it. Yes.
Are you drinking coffee? That's not coffee.
Yep. Copy right there. All right, Danielle's turn. All right, Danielle, what's the last kingdom then? It has mainly single-cell organisms in it, like the amoebas. Wait. Okay, couple seconds, otherwise you gotta go on.
I don't know.
You gotta guess? All right, Lana, let's look right around the room. Which one? Protesta is correct. It's correct. All right, should we write it down, Danielle, so that you won't make a note to study later? All right, head in the back. All right, whenever you're writing out the scientific name, which part goes first?
The genus?
Good job.
Write it on a 2.
Okay. Jaden's turn. What's the second part of the scientific name? So it's genus?
Species.
Good job. All right, which one's capitalized? Genus or species in the scientific name, or both, or neither?
Species.
Nope. Genus. Oh. All right, back over to you, Danielle. All right, which one is italicized? Genus, species, or both?
Both.
Both. Roll.
One.
All right. I'm sorry. All right, let's go over the classification system. So starting off at kingdom, what's the next one?
Phylum.
Good job, roll. Alright, kingdom, phylum.
Class.
Alright, you have a question?
No.
Or you're just ready? Okay. Kingdom, phylum, class.
Order. No, kingdom. Phylum. Class. Order.
Oh, you're right. You just psyched yourself out. Alright, kingdom, phylum, class, order. Family, roll. Alright, kingdom, phylum, class, order, family. Final answer? No. Kingdom, phylum, class,
Genus.
order. Genus. Alright,
Roll.
yep, because it rolled on
Two.
you. Two, I'm sorry. Kingdom, phylum, class, order, family, genus, species. That's the final one. Let's see. Alright, who's remembered as the father of the scientific method?
Bacon.
That's Bacon. This is what you were thinking of earlier.
7.
Alright, what is facts about nature?
Wouldn't it technically be observing hypothesizing?
Oh, no. What do you call just facts about nature? Don't worry about this yet.
Oh!
It's not your turn. You gotta wait till I go around the loop.
Wait, facts about nature?
Facts about nature.
What are the two
If you're observant, you'll know.
Observant.
No. That sounded so mean. No, please, we gotta go. Okay. Alright.
Observation.
Yep.
Data.
Data.
Oh, no, she was gonna get it.
Alright, roll.
Wait, was that?
Yep, it was a good roll. Alright, data. Alright, thinking about the general stages of the scientific method, what's the first general stage? Not the specific steps yet, just first general stage.
Observing.
Observing. Roll.
Done.
Alright, Jada's turn. What's the second? Good job. Alright, last major step or last major stage. Experimenting. Alright, now, what do you call a sensible explanation? Yep. Alright, what do you call a hypothesis once it's past your experiment and other people's experiment?
A law.
A law? Final answer? Nope.
What theory?
Theory. Alright, what's it called once it's passed a test of time now? As far as we know, it's never been proven wrong.
A law.
That's a law. I'm hearing lots of dice noise. Remember, he can hear it a lot better than I can. So make sure you're being quiet around the camera. Let's see. Okay, let's go through these. What is the first step of the scientific method? You might be cheating now, it won't help you later. Alright, Jaden.
Same problem.
State a problem for all. Alright, what's the second step? I'll take that. Research or gather information. Roll. Third step. Make a hypothesis. Okay, roll again, because that was a good roll. There you go. Alright, what's the fourth step?
Experiment.
You done? Yep, test your hypothesis. Alright, Lana, what's the fifth step?
Analyze.
Analyze the data. Jaden, you're about to lose it because you're getting distracted. Alright, and then what's the last step?
Report results.
But yeah, so state conclusions or report your results. Alright, we introduced these yesterday. There are 3 different types of variables you can have in your experiment. What's the one that you are looking at to see what changed about your experiment? Like, are the plants growing at different speeds? Or if our beans later, we're going to check to see if they start swelling up in water. So what do we call that whenever you're looking to see, okay, what's changing? What type of variable is that?
Wait.
We talked about this yesterday. It was really recent.
I mean, I understand what you're saying, but I don't at the same time. Like, I don't understand.
Okay, what's the variable that you're looking at at to see, okay, what's changing about my plants? Or what's changing about the beans. So it's what you're watching.
If the clay pot or the plastic pot affect how the plant grows.
Okay, that's your hypothesis. So how would you know, if your hypothesis was, putting them in different pots will cause one of them to grow faster than the other. So how will you know if your hypothesis is correct?
You do everything as almost exactly as possible as you can.
Mm-hmm. But what happened, but what are you watching to see with your plants?
If they grow faster than the other.
Alright, so you're looking at the speed of growth. So what type of variable is that? See, okay, I'm watching the speed of growth to see is my
Is it observation?
Okay, it's a type of variable. I'm sorry, we'll go over this later. Alright, you remember? What were we talking about? Remember, Lynn? Nope. Which one?
Independent?
Not independent variable.
Something variable.
It is something variable. Dependent variable. That is what you're depending on. We'll go over the terms again in a second. Alright, what is that one difference? Like in the potted plants, I had one made of clay, one made of plastic. What?
Independent?
Independent variable. That's that one difference. And then what do you call all the other variables that you are controlling to make sure that they're all exactly the same?
Well, the only thing I've heard is independent variable.
Well, we haven't said this one yet. But you're controlling them to try to keep them consistent.
Control variable?
Yes, roll.
Oh, I actually know that.
Alright, what do you call the groups on which you're actually experimenting with? So with the geraniums and the plants, I had 2 of these to test to see which one's growing faster. So what do you call those groups? Whenever this really fast towards the end of the hour. Alright? Danielle? Not control group.
I ain't got those.
Alright, Jaden, last one.
Repeat the question, you only said it once.
What do you call the groups to where you're actually experimenting with to see what's the difference between them? It's not control group, there's another word for them.
Differentiated groups?
Good guess, but no. Alright, everyone write it down then. Your experimental groups, because these are the groups you're actually experimenting with. We're going over this really quick towards the end of the hour, so we'll go over them again. It'll make more sense once we practice with the bean plants. Well, bean seeds. Alright, what is the group then that I'm going to use for comparison? This one is the normal one. That's a good guess, but no. Nope.
Can you repeat the question? I couldn't really hear it when you said it the first time.
Alright, what do you call the group that's used for comparison? It's the normal one. So, like if you were testing different types of soil, you would put if you're testing different types of fertilizer, you'd have one plant of regular soil and then fertilizer in other plants. But you would have that first one is the normal one to see, okay, here's how a plant normally does. That was it, so I gotta go. Alright, Lana? It's okay, I'm talking about it today.
A component group?
Nope, alright. Control group. Someone said this earlier. Control group. Write it down. Control group. It'll help you to study it later. Alright.
Every time the thing is done, it's finished on me and I'm not able to do another question.
It's okay. We'll try to do one more round. Alright, we'll go back to some easier questions now. Alright, what do you call a water environment? You should still be studying this stuff even though it's farther back. Alright, water environment. Couple seconds, Lynn. What? It's okay. Quiet, roll. Alright, do you have an example of a freshwater environment? Okay, what's freshwater? What's a body of water that doesn't have salt in it? Lake, pond, stream, river. Alright, cool. What do you call an animal that lives mainly in trees? Good go, roll. Alright, what do you call the area between the highest and lowest points on a tide for animals living?
Intermediate tidal zone or something like that. Intertidal zone?
Okay, now you have it, intertidal. Alright, Jaden, you ready? What do you call the hot springs in the ocean floor that help?
Hydrothermal vents.
Good job, roll. Alright, we made a lot of rounds. I'm sorry, Jaden. How many of you got 20 or more points total?
I haven't done.
I did.
Quickly. Alright, 20 or points or more. Alright, 30 or more.
I'm trying to add them up.
Oh my.
I just did mine in the little dashes.
Yeah,
That
so did
works. That works as well.
Hold on.
Couple seconds, add a point, quickly.
I can't think right now. I'm not in that class. Can't think. Oh.
Okay, you ready?
Okay.
Alright, who has 30 or more? Alright, who has 40 or more?
Okay.
Alright, who has 50 or more?
I'm at 46.
How many you have, Lana?
45.
Aw, you just barely won then. Alright, I will take the dice back. Alright, go ahead. Whoa, thank you. Alright, go ahead and get out your textbooks. Turn over to page number, I believe we're in 20 now. 24. Let's finish talking through this. Thank you. I will risk Corona. Alright, we talked through the first two already. Now let's go through the rest of the list. Now keep in mind, you don't have to memorize this list, so don't worry about thinking, do I have to memorize all of them? No. But we're just going to talk through some good things that a good scientist needs to have. Alright, I don't remember who we started off with reading with yesterday, so we'll just rotate and just start going through it again. Alright, anyone want to read the third one, orderliness? Alright, Hayden?
An effective scientist must be orderly in many areas, from keeping track of control and experimental group and experiments to recording, presenting experimental results, and conclusion.
Good job. So you have to be neat and organized. Now, you might think, but I'm not very neat. Okay, for your experiment, you need to be, or your demonstration. Whenever you are doing your experiment, if you're in 8th grade, you are going to have to be very organized. Make sure you're writing down good information. For 7th grade, since you're doing a demonstration more, you should still be orderly though, or keep your area organized. That way you don't lose something on your demonstration. We're on page 24, Jade. Don't worry, we'll get to that. Alright, who wants to read the next section? And the next one, intellectual honesty. Alright, Evie.
Intellectual honesty. An effective scientist should be honest about his own mistakes and about his observations that do not support his hypothesis. He also gives proper credits to the ideas and research of others that claim they're not his own.
Good job. So, intellectual honesty. So, if you realize, and you're in 8th grade, that your hypothesis is wrong, will you fail this STEM project?
No. Yes. No.
It's okay to be wrong. Now, she's in 8th grade, so that's probably the reason why she has to come up with a full-on hypothesis for hers. So, if you discover your hypothesis is wrong, I'm not going to fail you. It's okay. Scientists make wrong hypotheses all the time. The point is you need to learn something through the scientific method. So, don't worry about, it's wrong. This Morse is going to fail me. No, this Morse will not fail you. But if you realize I was wrong, it's okay. Just say you were wrong. Everyone's wrong at some point in their life, so don't worry about it. Now, your book also points out you need to give proper credit to the ideas and research of others instead of claiming them as their own. This is part of the reason why we practice writing essays, but then also what's it called whenever you steal someone's words or ideas? Mrs. Henderson has probably already talked about this in English class. Glenn. Plagiarism! Should you ever plagiarize someone's own works? No. Now, if you need to quote someone, put it in quotation marks, but don't ever say, I thought this up, and put it on a paper. So you want to avoid plagiarism as much as possible. I'm sure she goes over it with you much more than I do. But just make a note whenever you are writing your background papers later on for me that you don't try to plagiarize. Alright, next one. Who wants to read it? If you already had a turn, I appreciate it, but I gotta give other people a chance. Alright, dude. Acceptance of uncertainty.
Oh, I see it now. An effective scientist knows that a result of an experiment may not always allow him to draw a conclusion about his hypothesis. He will accept this uncertainty until the evidence
There you
allows
go.
making
So sometimes the scientists might have to test it over and over and over again. And that's okay. Don't just think, well, I've wasted my time. No, you need to do it over and over again to make sure you're 100% certain about your results. Alright, next one. Who wants to read? Danielle. Skepticism.
Skepticism. An effective scientist is skeptical of scientific ideas, both of his own and those of others, and will not accept them unless they are supported by sufficient evidence.
A lot of our ideas, laws, and theories today are backed up by scientific evidence. But if someone comes up and tells you something random, don't necessarily just believe it just because a friend told it to you. You want to make sure and go back and double check it, fact check it. Look, in some cases you might even want to test it yourself. Don't just believe it just because someone told you. Now, I, as your teacher, I've gone through and I have fact checked a lot of different things, and I found a lot of different other research for it. So I try to be as accurate as possible. So you don't have to worry about it with me. But if another friend just brings you something and just tells you something and it sounds weird, there's nothing wrong with fact checking it yourself. It's okay. Alright, last one. Who wants to read? We've already had a turn. Put your name down. Lana.
Openness to new ideas. An effective scientist is willing to listen to and carefully consider the scientific ideas of others. He does not reject others' ideas merely because they disagree with his own.
So because of that, you should still, even though it sounds like that the idea, it sounds weird, don't just think, okay, well, it sounds weird, I don't like it, I don't want to believe it. Okay, no, it might be true. Now, keep in mind, though, that there are some things that we as Christians always believe, no matter what the evidence seems to imply. What do we believe, no matter what man says? Alright, because this never changes. Jaden. The Bible. That's part of the reason why we talk about creation and evolution, because a lot of people believe evolution, and they say that they have evidence for it. Now, we'll look at it later on in class about the evidence and that they have. Their evidence actually matches up better with what the Bible says, but since they don't want to believe the Bible, they want to come up with something else to say how we all got here. So, just keep in mind, though, that we as Christians, we believe the Bible first, but then since God wrote the Bible and since God made the world, the evidence matches up with the Bible as well. Alright, any questions about this before we move on? Alright, so going back a little bit, going back a little bit with your variables. If you didn't mark these yesterday on page 24, go ahead and mark them. So a dependent variable, that's the one that you watch. It'll make more sense as we talk about it in a second. Independent variable, you can only have one. That's that one difference or that one change. Control variables, that's everything else in your experiment. The experimental groups, that's the one that you're actually working with. This one has the changes, while your control group, that's the one that's used for comparison. It's the normal one. So if you were getting kind of confused yesterday about it, make sure you mark them on the top of page 24. Alright, so your book gives you an example really quick in the tackle this, so we'll talk through this and then we'll work with ours. Alright, so suppose you design the following 3 experiments to record the growth of bean plants from seeds. All the experiments have identical control groups, and using the information below, determine which conditions are the independent variables and which are the controlled variables in each experiment. So we have that first one. It has the first one with the happy looking sprout of a control group. I'm going to write CG for control group. Alright, control group. If this is a control group, is anything going to be changed about it? No, this is the normal one. So for this one, it's planted in regular soil. I'm just going to write RS for regular soil. What?
The beans are its arms.
Yeah, that's its little like leaflet seeds. So we're going to place it in sunlight. So direct sunlight, I'm just going to write S for sunlight. We're going to water it every day. So water daily. Alright, so that is my control group. Now, I have the other experimental group. These are the ones that I'm actually changing something about to determine how well they grow. Now, listen to that first one, experimental group number 1. I'm just going to write all through the side, experimental group number 1. I'm going to run out of room, Josh, so I'm going to start writing in other places around it. So, experimental group number 1 for it. Alright, so this one is planted in regular soil. Is that the same as the control group? Glenn? Yep, so this one's planted in regular soil as well. This one is placed in direct sunlight. Is that the same as the control group? Edie? Yep, it is. In regular sunlight. But I'm only watering it once a week. Alright, is that the same as the control group or is it different? That's different. It's going to be watered once a week. So I'm just gonna write WW. That's what kind of he looks kind of sad and unhappy in the pictures because he's dying of thirst, basically. Alright, so comparing these two groups, what is that difference? What is the one difference in between them? Lana?
One of them is watered every day and one's watered once a week.
So what do you call that one difference?
Like, independent variable?
Good job, independent variable. So that's what it's asking. Asking for this tackle this. You're supposed to see which one is the independent variable and which one are the controlled variables. So out of these three, this is the one difference. So these others are the controlled variables. Alright, anyone have any questions about that first one over what we're doing, just having practice determining? Alright, let's look at the second one then. So that second one, I'm going to just erase it and I'm just going to change it to number 2 now. Alright, so it's placed in regular soil. Is that the same, Danielle?
Yeah.
Yep, this one is placed in regular soil. Alright, it's placed in the shade though. Is that the same? Evie? No, it is not. So we're just going to put it, I'm just going to write SH for shade. And then it's also watered every day. So is that the same, Jaden?
Yes.
Yes, it is. So water daily. Alright, so what is the one difference in this experiment comparison? Glenn?
Independent.
Okay, yes, it's independent variable, so you're right, that's the term. So what's the difference between the control group and this experimental group number 2? What's the one difference between them? Alright, are they both planted in the same type of soil? Are they both placed in sunlight? No, so that's the difference. Are they both watered every day? Alright, so what's that difference? Okay, you're right there. I think you're overthinking that. Yes, the independent variable for this one is that one's placed in sunlight, one's in the shade. And so you're right that that is the independent variable. All the others are the controlled variables. That's what comes in this picture. He looks kind of happy. Now, I don't know if the person actually put him like underneath an umbrella. It's just for the picture. Alright, did you do the question, Jaden, or were you just ready to answer? Alright, now looking at the last one, anyone think they know just what the independent variable is just by looking now instead of me talking through it? Alright, Danielle.
Cleaner in soil and fertilizer.
Yeah, because that's the difference now. Everything else about it is the same. It's placed in direct sunlight and it's watered every day. That only for that one is the first one is it's placed with fertilizer. So that's the difference between them. Anyone have any questions about the idea of independent variable now? Welcome, Scott. You made it just in time. Alright. Anyone have any questions about this before we move on? Yes, Jaden. Okay. Flip over to page 26. Designing an experiment. This will be our practice one for it. Alright, so we'll give other people a chance to read as well. Okay, we'll let Hayden go. Alright, Hayden, read the top part.
Suppose you're asked to design and perform an experiment to determine how water gets into seeds so they can begin growing. While carefully examining the surface of the bean's seed, you notice a small spot at one end of what appears to be a scar. That your hypothesis may be that the water enters the bean only through either the scar or the tiny spot. In this activity, you will perform one possible experiment to test this hypothesis.
Yeah, so we're looking at this to see, alright, where does water enter into the bean plant from? Now, I already have some beakers set up for it. I went ahead and I got water yesterday. So for this one, we're going to look to see with the different beans, where does it come into? And more specifically, does the little bean scar, that little spot on it, does it actually affect it? I'm going to give each of you a bean so that way you can look at it yourself. What they're talking about is a little white spot towards the center of the bean. That's where it's asking you, do you think that the water enters in the bean through that spot? So that's what you're actually looking at to test to see, okay, does it come into? So if you look at the bean, you will notice that it has this kind of this white mark toward it. These are pinto beans. We talked about this I think briefly yesterday. So if you look at it, you know, okay, that was kind of like a weird mark. So is that where water gets into the bean from? So let's say you have a hypothesis saying that, okay, if I were to cover that up with nail polish, that should keep water out of the bean then, if I think that that's where the water is getting into. Alright, so let's read through the next step. Who wants to read? Jaden.
Step 1?
Yep.
Select 12 bean seeds Those seed coats have no visible cracks which water would enter. Divide the seeds into two groups, 6 seeds for the control group and 6 seeds for the experimental group.
Alright, step 2. Alright, who wants to read it? Who hasn't had a turn yet, but I appreciate it. Scott.
Completely cover the spot and start heating the experimental group with nail polish. After washing the machine, place it on the dish, let it dry with a sharp cleansing cover. Do not touch the nail polish until it has dried completely. You will have them filled up to approximately 3 water.
Yeah, I went ahead and did that yesterday so that way it doesn't smell like nail polish in here. I actually used 3 different types of nail polish just to make it more interesting. Now, for one of them, I use the clear, nice, the top coat ones. If you're a girl and you paint your nails a lot, you know you're supposed to put on a top coat on top of it to help it last longer. So this one is supposed to kind of help cement it. This one is a good brand of nail polish. Again, if you paint your nails a lot, you know that LA Colors is kind of a nicer brand. It's a little bit more expensive. And then this one is cheap nail polish. Alright, so not only are we testing to see, okay, nail polish versus just the regular bean, we're also seeing, okay, what type of nail polish, if it keeps out the water, which one works the best? How many of you think that, how many of you think that the red nail polish will keep it out the best? Nobody? How many of you think the blue one will keep it out the best? Because it's a nicer brand.
And it's because my favorite color.
Okay, that works too as well. How do you think that the clear one will work the best?
Because it's supposed to help keep it out from the nail polish under.
Yeah, it's supposed to help it last longer. It's not gonna be always put on a top coat. And then how many of you think, I don't think it matters, I think water's just gonna get in it either way. Okay, a couple of you. So we're gonna test it to find out as well. Alright, let's look at step 3. I've already painted them like I said, so you can see the different colors on them, except unless you're Josh, because of the camera, and I'm sorry Josh, I don't think you can, I'm sorry. Alright, so let's move on to step 3. We're going to skip that one because I don't have a scale sensitive enough to pick up the bean weight because beans don't weigh a lot. So I don't have one small enough to pick it up. We're just going to use our eyeballs. Now keep in mind, in a real experiment, you would have to come up and actually weigh them or measure them with a ruler. You can't just say one got bigger than the other. But since this is just a practice one and we're just demonstrating it, and it's the one that your book talks about, we'll do this one. But just keep in mind that in real life, whenever you come up with a real experiment, don't just say, I saw with my eyeballs that it got bigger. Okay, no, you'll be able to measure it or determine it in some way. But we're going to skip that for now. Alright, so let's jump over to step 5. Evie.
First of all, I'm going to use about 2 cups of water into each of the 2 beakers, or measuring cups. Label one of the beakers for control group and one for experimental group. Then drop the seeds of each group into the appropriate beaker. Place the beakers in one location while seeds to separate.
Good job. Alright, who wants to help me? Alright, we'll take turns then. Alright, Hayden, you come up and you start. Alright, doesn't eat bucket. Oh, what? No, do not eat it. Remember science class, you never eat beans. Alright, I'm gonna slide it off to the side so that way you 2 people can't see you. Sorry, Josh, you'll have to. No, we're doing it correctly. Alright, so this is a beaker. Should you drop the beaker? Never, ever. What happens if you drop the beaker and it breaks? What are you supposed to do? Tell me, which I will obviously know that it is not broken. But make sure you tell me. Alright, so this is just water for you. So you want to try to get 150 milliliters. The 150 mark is right here. Just try to pull up to that one, okay? Don't get water on my electronic stuff. You pour it out. Okay. Is that 150? I keep the mark in there. Yep, good job. Alright, go ahead and pour it in here then.
Why don't you just use the little tip?
It's okay, you can do it however he wants to pour the water. Alright, which beans do you want to put in it? The red ones, the blue ones, or the clear ones? Red? Alright, here's your red ones then. Okay, now, I didn't want to do 6 because that takes too long for nail polish to dry and I was lazy. But how many should you do for each of your experiments? What's the magic number in science?
3.
3. Alright, here you go.
Nice.
Put your 3 beans in. This will be experimental group number 1. Alright, who wants to help me next? Alright, Jaded. I'll label this one experiment. Okay, stand over here so that way, because I'm worried you're going to get water on my stuff. No offense to Hayden. Alright, so 150. Again, 150 is about That is not 150.
It's like barely below.
Okay, well that's not the exact same, then let me get you a smaller cup so you can add slowly at it. There you go. Because remember, we have to keep everything else the exact same besides just having the nail polish. What do we call the type of variables if everything else, if you have to keep it the exact same?
Independent?
No,
Or
that's
something variable. Starts with a D.
If you're controlling.
Oh, dependent. No,
Nope.
control variable.
Control variable, so, everything is right. Alright, what color do you want? The clear ones or the blue ones? Alright. Alright, these are clear, they're shiny on the ends. Yep. Yep. This is why you have 3, in case one dies or gets broken. Alright, I'm assuming you were ready then to come help. Alright, come help me. Alright, how much water? 150, so it's this one right here. There you go. This is just regular water as a side note, there's nothing wrong with it. What you doing here?
He's acting like he's eating his bean.
Do not eat your bean. No eating beans in science class.
It looks so good. I keep peeling off the skin now, so I technically have another scar on it.
Yeah, that's how come the new book tells you to check to make sure ahead of time.
Yeah, I'm just gonna go with
Yeah, I'm
yours.
lost. That's what I've been doing.
Trial and error. If you want to put it on the counter and use a smaller beaker to add to it, you can.
That's what I did.
It's okay.
Hey,
You're not too loud. Remember, we can hear you.
hello, Josh.
Oh, there's another scar.
Alright, let me check to make sure. Yep, alright, let's go ahead and let's add it to the, do you want the blue one or do you want the control group? The blue, alright, here's the blue ones, right. Wait, let's add your water first. I know we're talking to you. Yes,
Can we actually see you?
can you, wait, can you see it?
No, can Josh?
No, I'll show Josh at the end, but because YouTube, I don't want your faces necessarily on it. I'll show Josh at the end, so sorry Josh, you're just gonna have to wait. Alright, you wanna tell me what the last one? Lana. Well, Danielle volunteered. Alright, Danielle, you can come help. Alright, last one. Now, I have to use this one for the comparison. This one doesn't have any nail polish in it, so 150. Now, if this one doesn't have any nail polish in it, so there's no changes on it, yep, that's it. Then what group is that if it's the normal one that's used for comparison? Jada?
Variable.
Alright, what group is it if it's the normal one?
Spend control.
Control group. So that one is the control group because we're using it to compare our results to. Yeah, you didn't know we're talking.
It's right above.
Yep, slightly 2 more, so you don't want just an itty bitty bit. Alright, let's put it down and let's add some more water to it this way. Make it a little bit easier.
It's exactly on the line.
Oh, perfect. Alright, so here's my last beaker. Alright, add your water to it. I'll go ahead and move it on this side. Alright, if I hear talking over there again, which I've already told you to stop, I'm gonna go back. Alright, and then here's your control group beans, because they don't have nail polish on them. You go. Alright, so now I will show it to Josh really quick. Yes, Jaden.
Can Josh hear if I whisper?
Yep. They can pick it up much better than I can. So I can hear you. So keep in mind that yes, you heard that. So make sure that you're being quiet. Even though, you know, I'm working with someone else in the front of the room, I still need you to be quiet so that way Josh isn't distracted. Alright, I'm gonna go slide it back over so that way you can see now, Josh. Alright, so this is our control group. these are our experimental groups. I know you can't see them very well, Josh, I'll put them off to the side for it. But we're gonna check tomorrow, because it really does take beans a while to determine whether or not they're going to swell. Now, how many of you, again, for your hypothesis, just for practice, how many of you say, my hypothesis that, my hypothesis that the blue ones will swell up the most? Anyone committed? No one's committed. Okay, Danielle's committed. How many of you say that the red ones will not swell? Okay, so you think that the blue nail polish will work the best, correct? Alright, so what should happen if the blue works the best to keep out the water? Should they be the largest or smallest?
They'd be the same size.
Yeah, they should basically be the same size. So, how many of you think that the red works the best to keep out the water? Okay, how many of you think that the clear one works the best to keep out the water? So that means that you think that these should be the smallest tomorrow. How many of you think it doesn't matter at all? I think they're all going to swell the same. So you're going to compare it to the control group and you think, okay, they're all going to be the same size tomorrow. Okay, about 2 of you. So you will find out tomorrow whether or not your hypothesis is correct. So what is our independent variable? We technically have 2 of them in this one. So what is an independent variable? What is our independent variable in our experiment?
The blue, red, and clear.
Yep, nail polish. So not only are we comparing the nail polish colors against each other, we're also comparing it to a normal one. So it's not just the nail polish, it's also the nail polish color. Now, for a good experiment, you only need 1, but not for this one. I'm comparing something and I'm comparing it to a control group. So that's how come for this one we're using it and we're being careful enough for it. So if you're going to have a 2-part independent one like that, you have to be super, super, super careful. But a typical experiment, you only need 1 at a time. Alright, so let's see, what is our dependent variable? What are we looking at to see what changes? Anyone think you know, Glenn? The size, so that's our dependent variable. And then all of our control variables is everything else. We put in the exact same amount of water, kept them the same type of beakers, we're going to keep them in the same area over there so they stay the same as much as possible. And tomorrow we'll analyze and look at our results. Alright, anyone have any questions about the experiment? Okay.
Whenever I walk out of class, am I allowed to eat my bean?
Nope.
It's not part of science.
Nope, it's still part of science, whether or not you're in the science lab.
Wait,
Nope, don't eat your bean.
No,
You want to
no.
throw away the bean, you may throw it away
What
out.
happens if I eat my bean?
Alright, how many of you mean? You have broken science laws. Rules.
Good.
I mean, you probably won't die, but you still should probably not eat it. It's not going to taste good. It's like eating a regular bean that's uncooked. No. No. You want to, if I see you eat it at lunch, I'll just stare you down. Just think.
Okay.
You won't die, but it's not a good idea.
Is there
Because
no-
what if you touch, because again, I cleaned these desks yesterday, and you've been handling the beans for like the past 10 minutes, so everything on your fingers is now on the bean.
Clean the
So
bean.
if you were to eat the bean, you have now eaten all the germs you have been touching all day. So it's probably not a good idea. Alright, let's go ahead and let's go over on page 28 while we wait for these to work and finish up tomorrow. Scientific reasoning. So we're getting into this section talking about logic. Now, we use the word logic super loosely, but in science we mean something a little bit more specific. Just make know that logic is the study of reasoning. So logic is the study of reasoning. It is essential to be able to use your head, use your head and use your brain to be able to figure out things about science. So this is the logic or the study of reasoning. A person that studies this is a logician, kind of like you're saying magician, but with lo in front of it, so logician, so it's a little bit different for it. Now, what they use facts already known about nature. That is our evidence as logic. Now remember, we also have another term for facts about nature. What is the other term for facts about nature? Anyone remember? Hayden.
Zoology?
No.
Animal? Farm?
Okay, facts about nature. Data. So for this one, if you're talking about it with logic, we use something a little bit different because they're trying to basically deduce or figure out something. How many of you have read Sherlock Holmes books before? I think you read a couple of short stories of Sherlock Holmes. I think maybe in your literature book, either this year or next year. I can't remember the exact year. I'll read a couple of short stories about him. He uses logic, or he used evidence to try to figure out or deduce what had happened. And in the book, Sherlock Holmes is always really good at it. Now, keep in mind that whenever you're trying to figure out something or use your brain to figure out something, The data that you know, or the data, or the facts about nature, you refer to it as evidence whenever you're talking about it in logic. So just make a note of that. It's a little bit of a different term. It's the same thing though. Now, they need to make a conclusion based on reasoning from evidence. So we call that inference. That's whenever you have figured out something. So a conclusion based on reasoning from evidence. So that's inference. You are inferring or figuring out something. So other terms are a little bit weird here. So make sure, inference, that's a conclusion based on reasoning from evidence. So you will eventually, once we finish our experiment, you will be able to infer or figure out, okay, based on what happened, was my hypothesis right or wrong? So we can do that, inference. It's a conclusion based on reasoning from evidence. Make a note of that, how's that sentence? What distinguishes scientific reasoning is that it uses empirical evidence from observations of nature, and that the inference is a hypothesis about how nature works. So just make a note for that. This is how we use this type of logic with science class. Alright, your book gives you kind of examples over how you use this every day already. So you know that, okay, maple trees, alright, think of it this way. Do maple trees lose their leaves in the winter? Anyone know? Sometimes in your head.
Yes.
Yes, they do. So if you have a maple tree in your yard, what's going to happen to its leaves in a couple of weeks?
0.5.
They're going to start to fall off. So
They
you
look
might,
really pretty.
what?
They look really pretty.
Yeah, maple trees, they typically turn like the bright red colors. So they're typically really pretty whenever they start to change colors. Now, you might think, but it's not fall yet. How do I know? Well, if you know maple trees lose their leaves, whether or not your tree has started to lose them, you know, okay, it will lose them in a couple of weeks because my tree is a maple tree. You don't have to know it right this second or see it right this second. You can figure out or use logic to know my tree is going to lose its leaves because it is a maple tree, and maple trees lose their leaves in the fall. So that's an example of using science and logic together to figure out something about nature. Yes.
I don't understand this picture.
Which one? We will get to it. We're on page 28.
Oh,
Stay with
let
me.
me know.
Alright, mark your terms. So, logician, these are people who study logic, make a note of them. There are two main branches that they use logic, or how they use these thinking skills. So, there is deductive reasoning and inductive reasoning. We use both of them in science, so your book is going ahead and just teaching you both of them. So, deductive reasoning and inductive reasoning. We're going to start talking about them a little bit more specifically tomorrow because I'm just about out of time. Just making sure you just make a note that there are two different types: deductive reasoning and inductive reasoning. We use both in science, so make a note of that sentence. Science requires both deductive and deductive reasoning. You are using both at the same time whenever you are working on your project. Alright, we'll go ahead and we'll stop there because I'm just about out of time. You don't have any homework tonight because it is Wednesday now. But make sure you are still studying, because have you had a quiz yet this week?
No.
No.
Only Wednesday.
Yeah, so just make sure you're staying on top of it, because that means you could have it tomorrow, or you could have it Friday. So make sure you are studying and keeping aware for all your turns. Like, thinking back over it, what do you call that one difference in an experiment? Yes. An independent variable. What do you call all the other variables that have to stay the exact same? Scott?